Today, I'm delighted to announce that WHO is in discussions with a consortium of companies and institutions to establish a technology transfer hub in South Africa.
Good morning, good afternoon, good evening, wherever you are in the world. Welcome to this scientific colloquium on enhancing mRNA vaccine production, coming to you directly live from Afrigen in South Africa. Welcome to all of you, and in particular to some of the ambassadors we have either in the room or online from some of the many countries who are supporting this. This is primarily designed, as I said, to be a scientific meeting. It's an opportunity for you all, and particularly for journalists, to have your questions answered about mRNA and about this hub program and what it's trying to achieve. But it's also very much about celebrating partnerships, and in this case, the partnership between Afrigen and the National Institutes of Health of America. The whole program really is about partnerships. There are many, many organizations involved in one way or another, countries that are behind this, countries that are going to be recipients through their spokes. So it's clear that something of this complexity can't be done by just one organization on their own. There needs to be input from a lot, because this is not just about developing a mRNA COVID vaccine. It's about developing a platform and then sharing that widely across low and middle income countries so that we are much better prepared for the next pandemic. So we have a very full meeting today, and there will be an opportunity at the end to ask questions. You can either do it by putting your hand up virtually, or putting your hand up really for those in the room, or by posting your questions in the chat, and then we'll assemble those questions and put it to our distinguished guests. So with that, we'll kick off, and I'd like to introduce first of all Claudina Lutz, who is the Director of Health Innovation at South Africa's Department of Science and Innovation. Claudina, the floor is yours.
Good day. Thank you for being here with us, and thank you for the repeat visitors as well. Enjoy the support. We really appreciate the support that you are showing to us. This is a joyous occasion where the long relationship that we do have with the United States of America on science collaboration can actually be demonstrated again. And I want to state that we are very proud to be associated with the mRNA hub in South Africa. And also, it is really heartwarming to see how the world has come together to support this effort. And what is, for me, the most important thing is we as South African scientists can demonstrate we can do it, but we're not doing it alone. We do it in partnership. And this is, thank you for the partnership that's being shown from the United States. Thank you.
Great. Thank you very much indeed, Claudina. And as you know, the support for this program is being led by WHO and the Medicines Patent Pool. And so now it's my great pleasure to introduce Dr. Samia Swaminathan, who is the Chief Scientist at the World Health Organization. Samia, the floor is yours.
Okay, thank you so much. I think the host has to...
You may be on mute at the moment, Samia. We just can't hear you. Can I just check with our technical colleagues that...
Can you hear me now, Charles? They unmuted me.
Yes, we can hear you now. Perfect. So sorry, Samia. Please go ahead.
No worries. Thank you so much. So good morning, good afternoon, and good evening, Excellencies, ladies and gentlemen. I just wanted to make some very brief remarks about the origin of this concept of the mRNA technology transfer hub and our journey in a relatively short period of time, which I sometimes still find amazing even myself. It all started with us, of course, observing in early 2021 the huge inequities in access to vaccines that we were seeing around the world, but also, of course, inequities that we had seen in access to other countermeasures like PPE and oxygen and even simple things like gloves and masks. And we saw that low-income countries were always pushed to the end of the queue. And this is something that had been observed earlier, and history was sort of repeating itself. It also became very clear that it was those parts of the world that did not have manufacturing capacity that were actually dependent on other regions. And for example, 99% of vaccines that are used in Africa on a routine basis are imported. And therefore, there's obviously this huge dependency. So while we were advocating for the equitable sharing of vaccines through the COVAX facility and also through donations, we also had set up a platform or ability to actually do technology transfer. We had the CTAP, the COVID-19 Technology Access Pool, through which we had requested countries as well as companies to share their know-how and technology. But then we saw that that also wasn't a hugely successful initiative. And so we felt we had to do something more. And so we decided that we would start this technology transfer program. And the aim was really to support manufacturers in low and middle income countries to produce their own vaccines and to make sure that they have all the necessary know-how and operating procedures to produce vaccines at scale and according to WHO good manufacturing practices. So in April of 21, we put out a call for expressions of interest to companies wishing to host an mRNA technology transfer hub, and I'll come to why mRNA in a minute. And by June, we announced, in fact, it was June 21st, I think, that we announced that WHO had picked Afrigen as the first global hub, along with the South African consortium, a very impressive consortium that had been put together. But this would be a hub that would serve all countries around the world, all low and middle income countries. An objective of this hub is to build capacity in LMICs to produce mRNA vaccines through becoming a center of excellence and training. Now, typically when one hears about technology transfer, it's usually bilateral. It's usually voluntary, it's bilateral from the originator of the technology to a recipient. In this case, a technology transfer hub, in contrast, is multilateral. It brings together in one single place all the elements that are needed to establish the technology. So you need obviously the know-how, the data, the formulas, intellectual property, the training, and then transfers the fully validated manufacturing process to multiple secondary users. And this is where the role of the medicines patent pool comes in as well, and their experience. And when is there a need for this? Whenever there is a request from multiple recipients to receive the technology. And when we see that there's limited interest in bilateral agreements is what we've been observing. And clearly there were many member states coming to the Director General and asking for support and help in making vaccines. So from the inception, the program has been designed with a medium and long-term perspective for pandemic preparedness. It is not a short-term program limited to COVID. I think we need to be very clear about this. Now, so far, we will learn more about the hub. We haven't received any technology from any established companies that have validated vaccines, but researchers using publicly available information have been able to generate a prototype vaccine that will now go into clinical studies. Now, we need a number of things to happen if this initiative has to be successful. The first is you need a workforce with the necessary skills and experience in manufacturing of biologicals. And this is why side by side, we have also established a biomanufacturing training hub in the Republic of Korea. But again, it's going to work as a consortium. There are other training sites around the world that will be used to train the workforce particularly from countries that wanting to establish facilities. And this is going to need a huge exercise over the next few years. We'll probably have to train people in the thousands, not in the hundreds, and they have to be trained across the spectrum of production of biologics. Secondly, we need strong regulatory systems in these countries. So there's no point building factories and establishing a production capacity if the regulatory agency is unable to provide the kind of oversight that's needed and to approve products, and if the community or the world at large doesn't have trust in those products and the quality. So this is why WHO, and it's on it from the regulatory side, is also supporting these countries to increase the capacity of their national regulatory agencies so they can approve medicines, vaccines, biologics, and they can be pre-qualified so that they can then be purchased by the international community. And we have recently come up with a roadmap for regulatory benchmarking, and we have maturity levels going from 1, which is absolutely rudimentary, to 4, which is of the highest standards. And there's progress. Many countries now are really gearing up and investing a lot in their regulatory capacity so they can become at least maturity level 3. As of now, we have 15 spokes around the world, 6 in Africa, 5 in Asia, 2 in Europe, and 2 in Latin America. And 5 of these recipients have already received the initial introductory training in Afrigen, and the remaining will also be coming in. There's already a rota that's been established, a roster established for this. Now, the reason, of course, we selected mRNA was because, well, the first is that mRNA vaccines proved so highly efficacious against COVID, and so far also against new variants. They're also easier to adapt for not only for new variants, but also for other diseases, other infections. And therefore, you know, this technology and training on this technology is likely to have long-term benefits for the regions where production will be based, because there is the potential, of course, to use this technology for many other vaccines and health products. It's also, though it's a new technology, it's relatively easy to share and develop, and the cost of goods will also come down. Of course, there's a lot of R&D going on around how to make them even more user-friendly in terms of being able to be stored at the normal refrigerator temperatures and so on. So there's already a lot of improvements which are happening, but of course then you need the lipid materials and so on. So one also has to think about the raw materials and components that need to go in, and where are those going to be manufactured. So there are a lot of downstream and upstream considerations here. So I'd like to end by really expressing my happiness and welcoming this formal collaboration and scientific partnership between the Vaccine Research Center and the NIAID of the US National Institutes of Health, and this technology, the WHO Technology Transfer Hub at Afrigen in South Africa. I'd really want to stress the importance of collaboration with centers of excellence around the world in order to achieve our objective of developing mRNA vaccines that are suitable for low and middle income countries. And I also think that this partnership could really also serve the needs of this network of scientists that are now growing, through this hub and spokes model. We've had enormous energy, enthusiasm from all of the countries wanting to be part of this global network, the scientists wanting to work together and collaborate. And I hope that this will become a very vibrant network in the future, really keeping up to date with the latest advances in technology and preparing ourselves for any future epidemics and pandemics, and also trying to address the unmet health needs that we see in populations around the world today. So I think this is a new model. We are just taking the first baby steps. We have a long journey ahead, but I see this as something that's that offers a lot of hope and clearly has generated a lot of interest already. Thank you so much, and wish this collaboration all the very best. Back to you.
Thank you very much indeed, Soumya. And I'd now like to hand over to Gray Handley, who's the Associate Director for International Research Affairs at the National Institute for Allergy and Infectious Diseases at the US NIH. to talk about exactly this collaboration between NIH and South Africa. Great.
Thank you very much, Charles. It's a real honor to be here and to represent the National Institutes of Health of the United States, of our Department of Health and Human Services, and in particular to represent the National Institute of Allergy and Infectious Diseases. Many people, and I am also bringing greetings from our director, Dr. Anthony Fauci. He is very much aware and very supportive of this partnership. It's an exciting partnership, a historic partnership, and one that all of our institute and our entire set of institutes is very, very excited to be a part of. Many people ask often, how did we get a vaccine that was so effective so quickly? It was because there had been over a decade of investment in all the basic research that was required to lay the foundation for all of the elements that turned out to be combined into an effective vaccine about when SARS coronavirus 2 emerged. We had done research on MERS, we had done research on SARS, and this was the foundation that we and our partners around the world, because it was an international collaboration, used to develop those vaccines that have all proven effective and saved so many lives. We see that as a model for the future moving forward, and we can only do it with partners like Afrigen and Biovac, where we can find top-level scientists and work with them in the future, and with the support of DSI, the South African Medical Research Council, and other longstanding partners. My task today that I was asked to do is to share with you some of that background information about how we got to this point so quickly and why we are so excited about the opportunities that face us moving forward. I think this will work. The National Institutes of Health, just for those of you who may not know, is the largest organization in the world that supports biomedical research. Our goal is to support research that leads to discoveries and the development of knowledge that will improve human health. The National Institutes of Health is actually made up of 27 institutes and centers. Many people don't know that. They do know about the National Cancer Institute, the National Library of Medicine, and now, of course, with COVID, you all know that the National Institute of Allergy and Infectious Diseases has a very large portfolio addressing infectious diseases all over the world. Not all of those institutes and centers are the same size. The National Cancer Institute is our largest, but NIAID, and this is without COVID funding, but NIAID is the second largest. And I think this demonstrates two things. It demonstrates the importance overall of research on infectious diseases and basic immunology and allergies as a continuing threat to human health. But it also demonstrates the overall commitment of the National Institutes of Health to international collaboration. Not all of the institutes contribute to international collaboration equally, but NIAID contributes the most. And that makes good sense because infectious diseases really do have to be studied in multiple countries in all the regions of the world, often in partnership with WHO and our other colleagues in the multilateral world, but primarily in bilateral relationships that we have with individual countries whose governments make an equal commitment to the commitment of the US government to advancing knowledge through the support of science. Try again. And that brings me specifically to the bilateral programs that we've had for many, many years with South Africa. For over 30 years, we've had research collaborations that cross the gamut of infectious diseases, as well as cancer, maternal and child health, mental health, and other chronic diseases. I've worked in a number of the other institutes, and I know just how important these collaborations with colleagues here in South Africa have been. We particularly have had longstanding collaborations with institutions that were traditionally strong here in South Africa, but we are working hard to develop relationships and expand those partnerships with previously disadvantaged institutions throughout the country. The University of Cape Town, the University of Stellenbosch, Witwatersrand, and many others have formed the foundation of this long-standing partnership, which has produced, if you'll review any of the literature, which has produced really world-changing public health impact literature and findings that have altered clinical practice and public health interventions that have been effective, particularly with HIV and AIDS, but also with many other diseases. This very successful partnership is really been formed because there have been unusual research opportunities here that we could explore together, but most importantly, because there are truly outstanding scientists who are very well trained and well supported by their own institutions. The NIH does, in fact, give out a lot of money, but we only support the research projects. It's fundamentally important that institutions like this receive support from their own governments and from other sources of funding, because that is the foundation on which we can build those research activities. Particular programs in South Africa that have been very productive have brought new technologies to South Africa using NIH resources. H3 Africa brought in technologies related to genome sequencing. DSI Africa is just starting now to be able to manage large data sets and analyze them for health impact and interventions. And the others that are listed there are some of the major networks globally that have explored treatment and prevention interventions for HIV/AIDS in particular. And one interesting side note about those networks, of which South Africa has representatives in all of the networks that we work on with HIV, is that when COVID came along and when we needed research sites around the world that were prepared to undertake clinical studies of interventions, both therapeutics and vaccines, we turned to this network and we asked those investigators to pivot away from HIV and TB and to undertake these trials, these important trials of COVID therapeutics, and they responded. Through those trials done in networks of investigators around the world, we were able within 9 months, and really now within 2 years, to develop and to discover and to evaluate effective vaccines and therapeutics. We see that as the kind of foundation that we'd like to build here, along with Afrigen and with our colleagues in South Africa at the funding organizations. One of the other things that makes South Africa quite unique in the NIH experience is that for the last 10 years, we've had this robust partnership with the Medical Research Council of South Africa. This is a unique partnership that does not exist in other countries around the world because of the vision of the MRC and their understanding that by working together, we could triple, not only double, but triple our joint research project. This is a jointly funded initiative. Both sides put in resources, both sides put in intellectual resources as well as financial resources. And over the duration of 2 cycles, we've had many discoveries and many advancements, which are quickly listed there. Now, within NIAID particularly, we're organized into some 7 productive, 7 programmatic divisions, of which one is the Vaccine Research Center, which is just highlighted there in red. We have a specific mission that reflects the mission of NIH overall, with our focus on infectious diseases, allergic diseases, and with basic immunology. But unlike the other institutes, which have similar mandates in their area of disease responsibility or organ categories, we have the responsibility to respond rapidly to new and emerging disease threats. People didn't quite understand what that meant until 3 years ago, but now I think everyone understands that the NIAID is the research arm of the US government that is charged with responding when we have a pandemic. Hopefully it'll be the only one that we all experience in our lifetime. But when there is a pandemic, we need to be ready, we need to move quickly, and we need to identify and work closely with our partners at WHO, the UN organizations, and particularly with our bilateral partners around the world. And to be able to do that, we need to form foundational partnerships and relationships on which those early and quick responses can be built. In our activities internationally, we seek scientific opportunities that really are unique. South Africa, Afrigen, and this partnership offers really quite unique opportunities to work together in a partnership and be prepared. We also contribute to the development of capacity, and I think you'll hear many things today about how this partnership will help enhance capacity not only here in South Africa, but in Brazil, Indonesia, Bangladesh, and other spoke countries and spoke institutions that will be part of this really quite inspired vision of enhancement of global capacity for vaccine development and research around the world. And we're very happy to be providing the technical expertise portion of that that we can from the United States. We also support many academic institutions. I know that the Afrigen already has a number of partners in our academic institutions who are funded also by us to support those engagements and partnerships. And then to be specific, I know many reporters like to see some numbers. The truth is that South Africa is by far the most important bilateral partner that the United States has around the world in terms of research investment. Almost 10% of the over $700 million that we spent in 21 on international engagement, about 10%, a little over 10% actually, was spent supporting investigators and institutions here in South Africa. That really does place us on a very firm foundation for this enhanced collaboration with Afrigen. Some of the activities that we were able to pivot in COVID-19 with partners here in South Africa are outlined on this slide. And many of them have led to changes in clinical practice, expansion of therapeutics available. And of course, this work continues even today as we study together the long impact and effect of SARS-CoV-2 infections, but also most importantly, as we continue to develop even more effective vaccines and track the emergence of variants, which we really will all be needing to address very soon. A couple of the special partnerships that have grown out of COVID-19 include the Ubuntu study, which some of you in this room probably were a part of, a continuing study of mRNA vaccines where variants are emerging, this being a particularly robust emerging vaccine environment, emerging variant environment. And then, of course, the one we're celebrating today, the Vaccine Research Center's partnership with Afrigen and BioVac. We're very excited about this. We're just getting started. You'll be reading a lot about it in the future, but we're very happy to have been asked to contribute the intellectual capital that we have, as well as to form a partnership as we work together to enhance mRNA capacity, not only to address coronaviruses that may emerge in the future, but also other diseases that are very important to us and to this region, including such diseases as tuberculosis, malaria, chikungunya, and even the ultimate goal, HIV/AIDS. This conclusion just brings together the thought that we're very appreciative again of being invited to be a part of this partnership. We look forward to how this partnership will develop. And we are most appreciative of all of the other partners who've come on board to provide financial backing for this program, particularly our colleagues in Europe and around the world who have been so generous in their support for this facility, which we toured earlier today, for the capability that will allow them to serve as the global hub for both training and for research, and ultimately, we hope, for the development of actual experimental lots of vaccines that we'll all be able to try out in some of our existing and other expanded networks. So thank you again for inviting us to be here today. And thank you particularly, Petro, for inviting the VRC to be your partner. We very much appreciate it.
Thank you very much indeed, Gray. And in particular, thank you very much for coming in person from the US for this meeting. And as I said at the beginning, this is a scientific meeting. And so I'm delighted now to invite Dr. Barney Graham, who is the former Deputy Director of the NIAID's Vaccine Research Center, to give the keynote speech today about mRNA vaccines. Barney, you have the floor.
Thank you. It's a pleasure to be here. It's an honor to be here.
Hang on just a minute, Barney. They need to fix the sound.
Dr. Graham.
Can you see my
on-
slides?
there we go. Now we have you. Fantastic. No, we don't have your slides. Can you share your screen?
Yes, we do.
Oh, okay.
Yes. Thanks, Barney. We see you and hear you, or see your slides and hear you. Thank you so much. Please go ahead.
Okay. Yes. Thank you. And it's a great honor for me to be with you. I wish I was there in person. To all the dignitaries and scientists and other partners in the room, yeah, my greetings from Atlanta, Georgia. I want to talk about some of the story of RNA today, but it's all based on the underlying premise that an infectious disease problem anywhere is a threat to public health everywhere. And that empowering and facilitating and sharing technology with regional scientists around the world to solve regional problems before they become global threats is should be a critical priority for all countries, high income or low income. And finally, the critical importance of basic research that underlies all the things that have happened over the last few years. I've got a few disclosures that I'm an inventor on patents, vaccine and monoclonal antibody patents for a variety of viral diseases. And I'm going to start by describing the Vaccine Research Center, which I left about 6 months ago. I'm still serving as a consultant, but it is where a lot of the work happened that I'm going to describe today. And it was started in around 2000 as a basic research facility to develop an HIV vaccine. But it's more than just basic research, and it includes uniquely process development, GCMP manufacturing, and a pilot plant. Dr. Jason Gall is running these two parts of the VRC, and he's present on the line today and is a key part of this collaboration that we're celebrating today. We can do phase 1 and larger clinical trials and do GLP analysis of clinical samples. And this system has allowed us to address things, even though we don't have an HIV vaccine yet. It's allowed us, the technologies that have derived from that have allowed us to address these many other problems, including emerging infectious diseases, using a variety of modalities, including nucleic acid vectors. And I'll tell you about both the DNA and the mRNA story. These products have been shared and distributed and researched around the world, and it's been done by this relatively small group of principal investigators and program heads who have been wonderful colleagues over these last 21 years and who I miss. We're faced with a growing public health burden of re-emerging and emerging viruses. And we have vaccine challenges for these and for other diseases that have unmet needs. And I think we've all recognized that the traditional public health approaches are effective, but somewhat limited. Fortunately, over the last 15 or so years, a number of new technologies have come on board that make vaccine development, it's taken vaccinology into a new era. And it's not surprising that new technologies open up new possibilities for vaccine development. This has always been the case. It allowed us to make influenza and yellow fever vaccines when viruses could be grown in eggs. And a lot of the live and whole inactivated virus vaccines that we have came because Anders et al. Learned how to grow, to make cell culture so we can grow virus at higher titers. And then came bioreactors and growing viruses in cells at higher density. And then the era of molecular biology allowed us to make recombinant proteins and virus-like particles and reassortant viruses and molecular clones. So every time new technologies arise, it allows new vaccine concepts to emerge. And with all these technologies that have come up really just over the last 10 or 15 years, we see this again. Some of these technologies help us make more precise vaccine designs or make more precise immunological measurements. Some of them, including the nucleic acid delivery that we're talking about today, help us go faster and be able to have a more rapid iterative design. And these correlate generally with the ability to be prepared or rapidly respond to new pandemic threats. And that is how RNA was so impactful during these last 2 years. The threats, the current threats and upcoming threats can be envisioned in a number of different ways, but I think we've seen over this last decade between chikungunya and MERS and Ebola and Zika, and now SARS, that we need to take a more proactive approach. And this observational study by Woolhouse, looking at new virus infections in humans during the 20th century, revealed an interesting finding that even though the number of new viruses were increasing in a linear fashion, the number of new virus families that cause human disease were beginning to plateau. And so there are about 26 virus families and about 120 viruses in those families that are known to infect humans and that we can identify as with a potential to increase human-to-human transmission and create pandemic threats. And so we proposed back in 2015 this concept of a prototype pathogen approach for pandemic preparedness. And we thought that developing vaccines for about 30 prototypic viruses through phase 1 and developing vaccine candidates for the other 90 viruses, at least through animal testing, was a large but tractable and finite project. And we thought dividing the pathogens into mechanisms of entry, as shown here in these 4 quadrants, could create working groups of scientists, pathogen-specific scientists, that could develop the basic research tools and information and reagents needed to do vaccine development for all of these viral families. And it could be supported by these core functions, many of which are available at the VRC, which allows us to do the kind of work that has been done. We've been responding to pandemic threats and trying to go faster since the beginning of the VRC. That is really critical even now for HIV, that we go as fast as we can. And a lot of our work has involved nucleic acid vectors, but DNA, which is something the VRC has become very adept at manufacturing, run by Jason Gall's group, and is the basis for the collaboration with the new mRNA biohub. And so from 2003 with the first SARS down to 2016 with Zika, we diminished the time of sequence identification to first phase 1 clinical trial from 20 months down to about 100 days. But to really be effective, we thought we needed to reduce this time even further down to the range of about 50 days. And to do that, and one of the reasons we felt that was necessary is because during Zika, we started thinking about how to respond to this, because we'd had experience with another flavivirus, West Nile. And we started designing a DNA vaccine near the beginning of 2016. And by March, we had a sequence we thought could be effective. And within 100 days, we're able to start a phase 1 trial. And within just a little over a year, started a phase 2b efficacy trial. But by the time we got there to sites in Central and South America, the major epidemic had waned. And if you cannot get an efficacy result during the outbreak, it makes licensure and product development and availability and marketing nearly impossible. So a lot of things happened during 2016. One was that during this effort, as we made a DNA vaccine, we also were testing mRNA vaccines made by a number of different groups, including Moderna. And during that time, we found that the mRNA technology had evolved to the point that it was much more effective than DNA vaccination, that 30 micrograms of mRNA was just as effective as 4 milligrams of DNA. So other reasons to go toward mRNA include the following, that as the mRNA is encapsulated in this lipid nanoparticle and delivers RNA through the plasma membrane, it is then a single strand of RNA that directly goes through the translation machinery to produce a protein, which is the vaccine antigen that is the target for our immunity. And it can provide very authentic antigen presentation. This will look just like the spike protein for coronavirus that is presented during the virus infection. It can induce both antibody limb of the immune response and the important CD8 T cell arm of the immune response. And for the CD4 T cells that help antibody, it is what we call Th1 biased, meaning that it produces a lot of interferon gamma, and we think that can make vaccines safer. The mRNA, the lipid, and the mRNA itself, the components are rapidly degraded, which we think makes it a very elemental vaccine and also very safe. Unlike DNA, which has to get all the way into the nucleus to be effective, RNA only has to be in the cytoplasm, which we think is one of the reasons it's more potent. Unlike poxvirus or adenovirus vectors, there's no anti-vector immunity, so you can use RNA repeatedly for many different purposes. And importantly, one of the themes of today's meeting is that RNA can be made by chemical synthesis. You don't need cell culture, you don't need bioreactors, you don't need all the big equipment involved in that. And it is a rapid platform manufacturing approach, even faster than the DNA. So in 2016, after evaluating mRNA, comparing it to DNA, we made an agreement with Moderna to do a pandemic preparedness demonstration project. This is adopted by NIAID. And in 2018, Dr. Fauci approved the demonstration project for Nipah. It's a paramyxovirus and also a pandemic threat. And so we made a deal with Moderna to work on Nipah virus. There was a big meeting in 2019 to trigger this demonstration project. And by the end of 2019, we'd already validated the manufacturing system and made a decision to use a small batch manufacturing process for phase 1. And this is important because in the long history of mRNA that was discovered back in the 1960s, one of the recent events back in 2017 was that RNA was being used for personalized cancer vaccines. And to do that, technologies and techniques were developed to make relatively small batches. So just a few milligrams could be made at a given time. For an individual with cancer, you make using their neoepitopes and creating a design, making it just for that one person, and delivering it back to the investigators within just a few weeks. That small batch manufacturing approach is what was enough for us to do a phase 1 clinical trial. And so that is what allowed us to go forward with this demonstration project. And then in 2019, a number of new studies came online in which RNA was being used for vaccination against infectious diseases. So the RNA technology developed over a long period of time and was just getting to the point that it could be used in 2019. The other thing that was happening, based on some of our work with respiratory syncytial virus, another envelope virus that has some properties similar to coronaviruses, we'd been working on coronaviruses triggered by the outbreak of MERS in the Middle East. And eventually we're able to, in collaboration with Jason McClellan and Andrew Ward, find the structure, the atomic level structure of a human beta coronavirus spike protein in 2016. And then we're able to show that we could stabilize this with just a few amino acid substitutions to prevent this fusion domain from refolding, rearranging. And at the same time, showed that this stabilizing mutation increased the protein production from transfected cells, which would make not only the preservation of these important vaccine antibody targeting sites at the apex of this molecule, but also improve expression of protein from nucleic acid vaccines that we were working with. So this collaboration with Moderna involved working on a prototype concept for both paramyxoviruses and coronaviruses, using Nipah as the prototype for the paramyxos and MERS as the prototype for coronaviruses. By 2019, it had progressed to the point that antigen designs were available for Nipah and for MERS that expressed either as mRNA or as a protein could protect animals against lethal challenge with either of these two viruses. And so, because of the prior work with the coronavirus spike structure and stabilization and antibody binding sites and mechanisms of neutralization, when this first report of a new respiratory disease came out, and on January 6th, we had heard rumors that it was probably a beta coronavirus, and on January 10th learned the sequences for that beta coronavirus. It allowed us to rapidly pivot because we had a pre-existing public-private partnership with Moderna to turn the demonstration project now from Nipah to the coronavirus. And so when the sequences became available, we could rapidly design reagents to make protein, develop assays, solve structures of the new coronavirus, start immunization in mice. Moderna rapidly used the antigen design that we suggested to make RNA. We received experimental batches of RNA by February 4th, got our first results in mice 14 days later, and within 65 days started a phase 1, and within about 6 months started a phase 3. You all know the rest of the story, that we had results for phase 3 within about 10 months from the time sequences were available, and then emergency use authorization within about 11 months. Now this is fast, but really in the future, we need to be even faster, because to really get ahead of these pandemics, I think we need to have something distributable to global populations within about 6 months. Since the vaccine has been available, this is data from Israel that rolled out the Pfizer vaccine in a very efficient, systematic way. And they showed that overall, even though it was implemented about a year after the pandemic started, that they reduced infections by about a third and reduced the potential deaths from this disease by about 2/3. So the vaccine was highly effective. In the United States, where nearly 1 million people have died of coronavirus disease, about half of those deaths occurred before a vaccine was widely available, and about half after it was widely available around April of 2021. And if you look at the deaths after vaccine was available, you can see in this graph that was published by the New York Times, but derived from data from the CDC, that virtually all the deaths, at least more than 90% of the deaths that occurred from coronavirus disease were in unvaccinated people. One of the reasons that people remained unvaccinated and have a vaccine hesitancy is that they were worried about how rapidly this vaccine was developed. It was a one-year story that things had gone too fast. As I've told you, this was really, could be described as a 3-year story of pandemic preparedness demonstration project and public-private partnerships, or an 8-year story of the antigen design, or a 15 or 20 or 40-year story with all these different types of basic research advances building toward the ability to do this in 2020. Most of this, I believe, was driven by our efforts to make HIV vaccines. And even though they have not been successful, much of the technology that was used during the coronavirus outbreak came from these efforts to make an HIV vaccine. And I really believe that continuing work on HIV vaccine development will solve many of the remaining problems we have in vaccine design and delivery. One of the great things that came from the pandemic response was that we now have this new modality of mRNA that is a way of delivering a message to cells that creates the protein antigen, that creates the immune response that we're looking for. And when RNA goes into a cell, it triggers a number of things. And so the types of things we need to do to design the mRNA that have evolved over these last many years include reducing the amount of double-stranded RNA, which can trigger these signaling molecules and TLRs that trigger innate immune responses and cytokine production, including mostly type 1 interferons, that can either cause side effects or limit the amount of time the cell can make the protein that is the vaccine. But during this outbreak, we've learned that RNA is safe and efficacious. Stability and supply chain is improving. It can be used as a small footprint, small batch manufacturing, which is well suited for low and middle income country manufacturing, and importantly, also support research with rapid iterative design cycles. And the research is important because RNA itself is not magic. If you don't deliver the right thing, the antigen design is critical. If you don't make the antigen in the right way with the right surfaces exposed, it won't make the right types of immune responses. So not only is RNA important for the research to get to the right antigen design, there's research on RNA itself that this is very nascent new technology. There's a lot of room for improvement in design and manufacturing approaches. So the RNA that was used by both Pfizer, BioNTech, and Moderna is basically a single strand of RNA with these different elements, all of which could be modified or improved over with new research. And you can modify nucleotides. For instance, the Pfizer, BioNTech, and Moderna use pseudouridine instead of uridine as one of the nucleotide bases, or 1-methyl modified pseudouridine. This reduces the amount of cytokines that are produced. You can also make self-amplifying RNA by using an amplicon segment from an alpha virus genome that makes a lot more RNA message, probably also induces a lot more of those cytokines, but it would be a race between protein production and cytokine production. There's things in codon modification, I think, that can be improved. We don't know very much about how RNA secondary structure and the algorithms for changing that to improve translation and improve the potential triggering of cytokines that could cause side effects needs a lot more work. The downstream processing, especially to remove double-stranded RNA, still needs a lot of work. And the formulation could also use work. But by formulation, I mean, how is the lipid nanoparticle made? And so far, over these last 20 years or so, people have found that the combination of these four different types of lipids, a cationic lipid, a cholesterol, pegylated lipid, and a neutral lipid, together create a lipid nanoparticle that can help protect the RNA and also deliver it to the cytoplasm. All of this chemistry of the lipid nanoparticle is still I believe, in very early stages of refinement. So there's still a lot of work that can be done to improve this technology. So in conclusion, I just want to thank all the academic and industry collaborators. This was an effort done by thousands of scientists and hundreds of thousands of volunteers around the world who helped make these vaccines come out so quickly for coronavirus. The NIAID, Dr. Fauci, and especially the divisions, the extramural divisions of DMID and the division of AIDS, who created this coronavirus prevention network, were critical to getting this done, and coming together of the Departments of Health and Human Services and the Department of Defense operations to create this, what was called at the time Operation Warp Speed, to support the rapid scale up and manufacturing of these products were also critical. And my academic collaborators at UNC and Vanderbilt were important. I want to thank the principal investigators that I spent these last 21 years with, the laboratory that I just left, and especially Dr. Kizzmekia Corbett, who was instrumental in the early days, especially of getting the mRNA vaccine concept through, facilitating and contributing to the Moderna product in particular. And Jason McClellan, who I've worked with on anything to do with structure. So we now have mRNA that's safe, effective, and scalable. It creates a lot of new options for research and development. The rapid design cycles allows, I think, local regional investigators to start solving their own regional problems before they become global problems. The chemical synthesis means it's simpler and is more likely to be set up with a small footprint type of facilities. It still has a lot of complexities, but it is simpler than setting up the large bioreactor systems that need more space and more equipment and more people. And the mRNA that we have now makes this prototype pathogen approach to pandemic preparedness feasible because of its ability not only to be effective, but to be rapid. And finally, I think we all need to work on the problem of access and uptake. To really achieve global public health, this technology needs to be accessible and products need to be available around the world. If we could have immunized the world in 6 months instead of what's probably going to be 6 years, we probably wouldn't have had these waves of successive new variants that are making our vaccines less effective. So between the access and the uptake, which is going to require a lot more effort and communication, transparent communication with the public so that they have a better understanding of biology and understand how valuable some of these new public health tools are. These are the things that I'm going to spend a lot of my time on moving forward. So I'll stop here and take questions if there are any. Thank you very much.
Barney, thank you very much indeed for that great talk. We will, if that's okay with you, leave questions to the last part of the session. But thank you again very much also for your ongoing help and support to this program. So I'm now going to turn over to Professor Patrick Arbuthnot, who's the Director of Antiviral Gene Therapy Research Unit at Witwatersrand University and the South African Medical Research Council. Patrick.
Thanks, Charles. I'm going to summarize in the next 5 minutes or so the work that we've been doing with Afrigen. We've been working very closely with them for about a year now, and our focus has been on the nuts and bolts, nitty-gritty detail of trying to develop the technology required for mRNA vaccination here in South Africa. As you just heard, we from the Antiviral Gene Therapy Research Unit, so we focus a lot on using molecular biology, applying it to medical problems. And previously we were working on trying to make new therapies, particularly for hepatitis B virus, which is a very important health problem. And of course, when the pandemic struck, it made sense. And in fact, it was incumbent on us to repurpose the work that we were doing to try and assist with developing mRNA technology in South Africa. And it was then that we established this partnership with Afrigen to try and improve the capacity of mRNA development and vaccination technology in South Africa. So just a little bit of background for those of you who are not familiar with gene therapy, basically it's work with DNA and RNA. Those are genetic materials, so hence gene therapy. So there's a very good fit between gene therapy and vaccinology. So the point of departure really was to try and make a vaccine against COVID, which we would use as a reference for further development of vaccines against other pathogens, and that's been made reference to before. So we really had to try and trawl through the available information in the public domain to try and get together what is required to make a COVID vaccine. And that was not particularly straightforward because the information is not complete. But I was thinking about this while sitting here next to Glenda Gray. We were contemporaries at medical school at Wits, and something that stuck with me from our student days, I'm probably the same with you, Glenda, was that we were always told that most patients in South Africa live in resource-poor settings. So you have to use your clinical acumen and try and work out what is wrong with the patient using incomplete information. So I thought of that when we were trying to work out what is required to make this COVID vaccine. So we took all sorts of bits and pieces of information, put it together, and we came up with a plan, and we implemented it. This was, I guess, about 9 months ago. And so what we did was we designed this vaccine template, and then we made pieces of DNA that were put together to make what is required to transcribe, which is the process of making mRNA in a test tube. So we put this all together. We made these plasmids, they call, these are the templates of DNA for mRNA synthesis. And we generated the RNA, and then we needed to test it. We needed to make sure that it wasn't degraded because, as you probably know, mRNA is highly unstable, which is why it needs to be stored in very cold temperatures. So we did all that kind of work, making sure that it was stable, not degraded, and that the quality was good, etc. And this was quite a lot of work to do. And then the next step was, of course, the formulation. And Dr. Graham referred to some of the components of these formulations that are used to make these mRNA vaccines. So again, we didn't have complete information, but we used what we had and we put it together. And we started a study in mice about 6 weeks ago. And we took these formulations and put them into the mice. And we measured the immune response to this vaccine, obviously the antibodies to SARS coronavirus 2. And of course, we used a spike protein to a messenger RNA encoding the spike protein, because that's obviously a well-characterized good immunogen to counter SARS coronavirus 2. And we, lo and behold, we got a very good immune response. We were very pleased with this. And then that was just evaluating the amount of antibodies to the spike protein itself. And more recently, in fact, yesterday only, we got the results of the so-called neutralization assay. So what that assay tells you is whether or not the antibodies to this vaccine are capable of stopping the virus from infecting a cell. And these antibodies that were produced in the mice were extremely effective. And I'm using the terms actually of our colleague who did the immunoassays, and that's Penny Moore. She says it's extremely high, this neutralization efficacy. So we were very, very pleased, are very, very pleased that this vaccine prototype that we've worked on is working so effectively. And I think that this has buoyed us, and we're very enthusiastic about pursuing this very vigorously. And I think this is a very significant milestone, and it's really coincidence that we got the results that we did yesterday and that this meeting should be taking place today. But I think that we are positioned in a very good place at the moment. I think that it's true to say that the technology development is the most difficult part. We no doubt have further challenges, scale up and meeting regulatory requirements, etc. But I think that we've overcome a very, very significant hurdle, and that we are in a position now to capitalize on that and take this technology further. And what we are, of course, focusing on as well, and this has also been mentioned before in some of the presentations, is that we're working on addressing other important pathogens in South Africa, TB, HIV, etc. They may be a little bit more difficult, or probably considerably more difficult than SARS coronavirus 2, because the spike was an easy immunogen to use. And the other important thing to say is that I think it's true to say, although we've had this success, I think that this capacity in South Africa is still a little bit thin. I think there's a lot of scope to build this basic molecular biology that underpins this very important technology. There's also a very exciting technology, and I must give credit to my two young colleagues, Abdullah Eli and Christy Bloom, who are young researchers who've taken to this work like ducks to water, and they have immersed themselves completely in it and being vital in the engine room of this, this work that we've been doing. But it's also very, very satisfying. I think that this work is really, as I said earlier, it's given us a lot of enthusiasm, and it's a good sort of training ground for young up-and-coming scientists who would find this, or they do find it, I know for a fact, they find it very stimulating that we can do this kind of thing, and the confidence has contributed significantly to that. So that's something which we're focusing on as well, to make sure that we build capacity. Other things as well, we need to make sure that we have the freedom to operate, that we don't infringe on patents. We are trying to develop our own intellectual property to make sure that we can develop products that can be commercialized, so that the whole venture can be more sustainable. That's quite an important thing. We need to be sure that in the future we are able to sustain ourselves. Thank you very much.
Thank you very much, Patrick. And that's great to hear that the program is so on track with those results. Fantastic news. And I'm now going to hand over to Dr. Karin Bock, who's the Acting Deputy Director of Pandemic Preparedness and Emergency Response at the VRC at the NIAID. Karin.
Oh, there you go. Hi.
Yes, we can hear you now. Great, thanks.
I couldn't unmute myself. Thank you. It's really an honor and a pleasure to be here. I'm assuming I'm sharing my own slides. I can do that. Okay. I'm having technical difficulties. Okay, it doesn't matter. A lot of the slides were
You can
already
share them here.
shared, but yeah, if you can share them on the other end, yeah, that'd be great.
We have them up.
Oh, perfect. So, like I said, it's an honor and a pleasure to be here. This is a very important collaboration for us. If you can go to the next slide, you'll see that pandemic and epidemic preparedness and response. I'm hearing an echo, sorry. Has been a central objective for the VRC. And we've been over the last 20 years trying to get better and better at it. So we hope that this collaboration is going to take us even further. So next slide. As Dr. Graham explained, we're adopting his priority prototype pathogen approach to try to address all the viral families with pandemic and epidemic potential in humans. So this is exactly the way the VRC is prioritizing and addressing our own pandemic preparedness plan. And this has been adopted also by NIAID. And it was already explained, like I said, for Barney. So what I really wanted to do is tell you why this collaboration is so important to us. And in the next slide, you'll see that we have many core competencies, things that we're really, really good at, and that we add to the development of medical countermeasures such as vaccines and monoclonals. But the most important one for today is that we have our largest research team really dedicated to process development and formulation innovation. So we do our own process development and formulation innovation, and this is what we bring to the table and what we can help during this collaboration. Next slide. Dr. Gold will go into more details about our plans to work together with Afrigen. But as you see here, we have an example of a very healthy pipeline that we have currently, we're currently developing at VRC. But my central message is that we hope that we, both Afrigen and VRC, grow together in our pandemic preparedness through this collaboration and trying to develop process development for both of us. Thank you.
Dr. Jason Gold, who's the Director of Vaccine Production and Strategic Planning at the VRC. Jason. Jason, are you there?
All right, there we go, I think. Can you hear me?
We hear you now.
Fantastic.
Yep,
Can you see my slides?
No, not yet.
I am sharing them.
Yeah, okay, they're up now.
Fantastic. Well, it's wonderful to be able to be a part of this colloquium, as well as the collaboration, of course, with Afrigen. And you've heard a lot of the science, a lot of the collaborations that NIAID has with entities in South Africa from Gray. And I'd like to shift the emphasis just slightly over towards how the VRC approaches industry collaborations. And that is something that's been a focus of mine since I joined the VRC back in 2013. And it became recognized as a key aspect for moving technologies from the benchtop into the realm of public health, because ultimately it's our colleagues in industry that bring these to market in broad availability. So without going into much detail around the VRC's scope, this is kind of building off of Karin's second to last slide of our research portfolio, which unfortunately I guess most people weren't able to visualize on the virtual meeting, but we have a broad portfolio, and this is supported by a lot of underlying technologies. And within the VRC, we're fortunate here to have a number of capabilities that can allow us to go from benchtop discovery all the way to evaluation of the clinical trial samples from the clinical trials we conduct with material that we produced in our own pilot plant. And in any of these stages of development here, very early stages of development, we can collaborate on a scientific level with industry. And we have done so, with the most recent example here actually being with Afrigen. And within just a little more granular detail, that, as Karin mentioned, we have an extensive process development program within the VRC. It includes over 100 scientists focused solely on process development, cell line development, which doesn't apply to mRNA vaccines, but it does apply to the other technologies we use here. And formulation development has been discussed briefly in the meeting so far, and all the analytical assays required for us to understand the products that we're making. So within the VRC, kind of along the continuum of vaccine evolution, we have been witnessing the progression of technology supporting these different vaccines and implementing them at the VRC. Each of those with a red circle around them denotes VRC capability. You may notice that in the category of RNA vaccines, even though there are approved vaccines, the VRC as of yet has not implemented mRNA LNP technology, but that's what we're interested in doing now. Now, following the prior model we were using was to access, as Barney alluded to, mRNA capabilities through collaboration with industry partners. And that was effective. And the interest in mRNA vaccines has gone up. And so we have made the decision internally to start bringing in industry relevant processes for mRNA and LNP production. And that's where this collaboration with Afrigen comes in for us. Now, over the years, we have learned to, as initially a more academic, research-focused organization focused on HIV vaccines, to a little more of a product focus. We've learned the value of interacting early on in our potential product development programs with industry partners. And this graph is just to illustrate that in the areas where we have emphasized industry partnerships, it's resulted in out-licensing of VRC technologies into potential products. It's not a one-to-one ratio, as you can see here, and there's examples where we don't have industry collaborations and yet we have licensees, and vice versa as well. But it illustrates that the VRC over the last 10 years or so has really looked at how we can use our discovery, antigen design, and manufacturing capabilities to be in alignment with potential products that can go into the realm for public health. Just to wrap up, long before we were collaborating with industry, there was a lot of collaborations with academic groups. And we just kind of took that baseline and modified it a bit to understand what makes a potential product program feasible and attractive to industry partners. There's more than one way to measure success. There's a couple of bullets here to illustrate that over the last 10 years, we've gone from one and then two commercial licenses for VRC technology to well over 30, which is, I think, a nice metric. But perhaps even more importantly, is that 4 United States FDA approved products are based on VRC technology today. Now, we do have this broad portfolio, which includes a lot of industry partners. However, Afrigen is our first South Africa industry partner, and we're thrilled to be working with them, particularly on an area as important as COVID vaccines initially. But really, when you have scientists in the room, as soon as we said, yes, this is a good idea, let's work together on this, we started looking at what are the other opportunities for us to work together and expand upon beyond COVID vaccines. And that's where we're, in addition to the COVID mRNA vaccine technology, we're really excited about what we could do in the future. Thank you.
Thank you very much indeed, Jason, for that. And you mentioned licensing, and so it would be remiss of me not to take this opportunity to thank NIH for the licenses that you've given to the Medicines Patent Pool for the WHO's CTAP. So I'd now like to turn to Dr. Karen Feder, who is the Scientific Director at Afrigen here. Karen, the floor is yours.
Okay, thank you, Charles. I'd like to say a warm welcome to Afrigen, to all our special guests who are here with us and those that are attending virtually. It's a great honor for us in Afrigen to enter into this partnership with the National Institutes of Allergies and Infectious Diseases of the NIH, but working more closely with the Vaccine Research Center. And although this is the official announcement and the celebration of the collaboration, our teams have started working together a while ago, and we've had multiple engagements. From a technical perspective, this partnership has significantly fast-tracked our vaccine development program. So as many of you know, in the absence of a turnkey technology transfer, meaning a full transfer of the mRNA COVID-19 vaccine technology, we sought technology partners very carefully and very strategically. And there are now a few, as the VRC, that are contributing to specific aspects of the mRNA vaccine manufacturing process. So with the VRC, in particular, their vaccines clinical materials program, the VCMP, and the platforms that they have established there, which actually serves the VRC to provide support for their preclinical proof of concept and efficacy studies. Afrigen, which represents the hub, has been given such great support and priority, having this project treated as though it is an internal project. So the VRC will be providing the starting material, the plasmid DNA, for our mRNA drug substance manufacturing, which is also coupled to a technology transfer and training, which is really critical for our staff. And it will enable us to establish the process in-house. It will also be very applicable for the vaccine product pipeline, which will also be using the mRNA platform. So I'd like to quickly just summarize the goals of the project. It is to advance COVID-19 mRNA-based vaccines and mRNA vaccines for other disease targets, to demonstrate the suitability of the NIAID's plasmid DNA process for mRNA drug substance manufacturing. And as mentioned earlier, technology transfer and training, and then the aspect of lipid nanoparticle formulation development and transfer. In addition to this, the collaboration will grant us access to valuable immunogenicity targets, which has come from years of collective R&D at the NIH, and has significant relevance for the burden of disease in Africa. And as we heard previously, these include Ebola, HIV, malaria, measles, and other vaccines. In turn, Afrigen, representing the hub, will contribute expertise which we've gained in mRNA manufacturing processes and LNP formulation capabilities. I'd like to now quickly give you an update on the mRNA hub's progress. So we continue to work very closely with our university partner, my colleague sitting right next to me, from Wits University, But the work in Afrigen has centered around scaling up the manufacturing process and the good manufacturing practice readiness within Afrigen. You've heard from Patrick Arbuthnot that we have confidence in our vaccine candidate, that it is in fact immunogenic and capable of neutralizing SARS-CoV-2 virus. Simultaneously, we have begun the training program for the various LMIC spokes. We have trained 5 spokes, including BioVac, which is also part of the South African consortium, and we will train another 4 before closing 2022. In addition to this, the local innovation network, which is going to provide products for our pipeline, which is managed by the South African Medical Research Council, has already started its program. And I think, as Patrick mentioned earlier, there are 3 main priorities for this program, and that is to address freedom to operate, meaning creating our own IP, new innovations, reducing the cost of goods of the vaccines, and addressing thermostability of the vaccine, making them more applicable for low to middle income countries. Lastly, I'd like to say thank you to all of those who have been involved in enabling this collaboration. We are looking forward to working together towards a common goal, which is improving and securing global health for all. Thank you.
Great. Thank you very much indeed, Karen. And as you mentioned, this is also about research. So I'd now like to turn to Professor Glenda Gray, who's the President and CEO of the South African Medical Research Council. Glenda.
Thank you. It's actually wonderful to be here. Our relationship with the VRC goes back with Bonnie Graham, even before he joined, when he was still at Vanderbilt University. So I've known Barney for many years, over 20 years, and we've worked together on HIV vaccines. And so I want to acknowledge this long relationship, as well as the long relationship with NIH and with Gray when he was here as the health attaché many moons ago. Yes. So it's wonderful to see that this collaboration and trust continues. The collaboration, the teamwork, and the bilateral arrangements are imperative to the success for South Africa. And this new establishment with the VRC is catalytic for the kind of work that we're going to do here. So I'm very excited that this is happening. Why are vaccines important? Okay, so vaccines are important because they prevent millions of infections at a global level, as well as save millions of people's lives. And so it's critical for us if we want to control, eliminate, or eradicate a virus or an infection. Vaccines are one of the most important tools to do this. And so the commitment of South Africa to this is important for us to develop our own capabilities. Why do we like the mRNA platform? It's because it's a very versatile platform. We've heard over and over again that this is a versatile platform. One of the biggest obstacles for access of new vaccines is the ability to do rapid deployment and scale up. And so the mRNA technology is that it'll enable us to be able to scale up rapidly and deploy where these vaccines are needed. They have the potential to be very inexpensive and incredibly scalable, as we've heard before, and eventually stable, hopefully. And that's wonderful about innovation, is that we start off with vaccines that require -70, then -40, and then refrigeration. And so I look forward to that development in South Africa. We do want to use mRNA technology beyond COVID-19, and I see the vision for HIV. Our collaboration with the VRC spans many years, and I'm kind of one of the oldest people in the room when it comes to HIV vaccines. So I've run phase 3 clinical trial after phase 3 clinical trial, and I'm still working and still believe that we can find an HIV vaccine. But also it's important to look at other causes of morbidity and mortality in South Africa. The MRC's job is to fund and conduct research that changes the lives of South Africans. And we do this by looking at the 10 most common causes of mortality, of which HIV and TB are one, as well as SARS-CoV-2 now. And we will work with the NIH to ensure that we can help contribute to vaccine development that has been largely neglected, and that includes TB and HIV. Notably TB pipeline is very small, and hopefully this contribution, this mRNA development can really spur on our ability to find a TB vaccine as well as an HIV. I look forward to our commitment towards malaria and contributing to other kinds of illnesses that affect low and middle income countries like dengue, chikungunya, and Zika. So we look forward to those kind of collaborations as well. So why is South Africa important? South Africa is important because we have great clinical trial capability. As was demonstrated during SARS-CoV-2, we were able to rapidly use our clinical trial sites that were developed by the NIH, 34 of them, into COVID vaccine sites. And these sites enabled us to evaluate the 826 J&J vaccine and eventually rapidly deploy that to the 500,000 doses that we got to give those vaccines to healthcare workers in South Africa. Again, another collaboration with the NIH. I know I sent Gray lots and lots of letters. The US government co-owned these vaccines with J&J, and they cooperated to get these healthcare workers in South Africa a much needed vaccine before the next wave. So we really are grateful for that. Not only do we have great clinical trial capability, we've just heard of our vaccine platform capability, and I'm glad that MRC funded my colleague at university, and there was no conflict of interest there. So it's just wonderful to see the capability of South Africans who stay and commit to developing South Africa. So not only do we have wonderful vaccine platform capability, but we also have an incredible laboratory infrastructure. And we've mentioned Penny Moore, Lynn Morris, Caroline Williamson, people who are able to not only who are able to compete at a global level with the immunological and virological studies. So we have wonderful laboratory capability that enables us to do both clinical trial research and evaluate them as well. We've mentioned also our wonderful regulatory capability, and very often we can submit our dossiers and our protocols to South Africa, our SAPRA, without having to go to FDA or EMA. So I just want to acknowledge that we are able to do this because of our great regulatory and ethical trial oversight. We've also mentioned the issue of training, and the MRC is committed to train the next generation scientists and manufacturers so that we're able to have a vibrant program here in South Africa. And already we're working with public and private partnerships to develop a 100 million Rand training program that will enable us to scale up those thousands of people needed for clinical trial manufacturing in South Africa. As we speak, we are designing the first mRNA SARS-CoV-2 vaccine trial, which we hope will start early next year. And so we have a vision that we are going into, as soon as we have GMP material and our preclinical work is safe and shows that the product is viable, we'll start our phase 1 trials in South Africa. By 4 p.m. today, you will get the first trial design. I spent the morning with my team designing the phase 1, phase 2A protocol, which will look at dose finding, safety and immunogenicity in both vaccine naive, because there are lots of vaccine naive people in South Africa, as well as people who have had a primary series. And so we'll be able to evaluate what the best dose is, how this vaccine looks in vaccine naive and in vaccine experienced individuals, as well as HIV infected individuals. We know that there are up to around 8 million South Africans that are HIV infected, and we know that the vaccines that we have are not optimal for their in terms, particularly if they are immunosuppressed and are not aviremic. And so we will also be making sure that the vaccine that Afrigen, the Afrigen mRNA vaccine, such a nice name. The African mRNA vaccine will also be, we will evaluate it and make sure that we can also deploy it to HIV infected individuals in South Africa. And so we're very excited about this. And we also look beyond HIV. We are a country that has both communicable and non-communicable diseases. And I look forward to us also looking at the use of this vaccine in cancer and other immune-related diseases that are caused by viruses. And so this is a great collaboration, and it's just wonderful to see this bilateral agreement with the USA flourish. And I'm just also very proud of Afrigen and the work that they've done in record time to do this. So I want to congratulate Afrigen, and also I'm very happy that there is this VRC Afrigen collaboration. So well done and congratulations to all of you. Thanks.
Thank you so much, Glenda. And now, finally, before we move to the Q&A section of this, and please do put any questions you have in the chat, or wait and raise your hand, but you're more likely to get them answered if you put them in the chat so that I have them in advance. I'd like to turn to our hostess, Petra Terre-Blanche, the CEO of Afrigen. Petra.
Thank you very much. Glenda, you're putting us on the spot here. I saw Karen's eyes going quite big. It's fantastic. So this mRNA Hub program is built on partnerships. And today, again, demonstrated that. We have so many of our partners here today in the room participating and contributing. I want to make a comment about the specific celebration we're having here today in this public-private partnership between Afrigen representing the mRNA Hub and the NIH VRC. This is a high-impact partnership in the short term, contributing to the timelines for us to get GMP material ready for the clinical trial that Glenda has designed in a record time. But it also is high impact in terms of the long-term vision of building centers of excellence, a network of excellence that can rapid respond to pandemics, but can also develop a pipeline of mRNA-based vaccines relevant for the burden of disease in low and middle income countries. Gray, you said thank you to us for partnering with you. We were connected. Glenda, you remember in the first meeting, Sophia, thank you. And from the first moment when we indicated that we need your help, it was amazing how the NIH team, Jason, thank you to you and your team, just jumped in with enthusiasm, working with us on designing this program. Thank you for that. And I want to say to Barney, I wish you could see the face of the mRNA Hub scientists sitting here to the left of me while you were talking. It was for them amazing. Even Karen's face. Thank you for contributing and sharing today, and the invitation for you to visit still stands. Thank you to all our partners, and thank you for the contributions today. And we look forward to working with the NIH for many, many years.
Great, thank you very much indeed, Petra, and thank you so much for hosting us all here today. So we're going to move to some questions. So first of all, I'd like to ask Donato from the Financial Times to put your question to whoever, if you could single out someone to put it to, otherwise I'll ask any of our speakers who want to answer. Donato.
Hi, can you hear me?
Yes, thank you.
Congratulations on this announcement today, and thank you so much for the briefing, and thank you so much for taking my questions. I have a couple. So the first ones are perhaps for NIH representatives. I'm wondering how much, if at all, the patent dispute with Moderna has anything to do with this. Obviously, we know the NIH has been deeply involved in both the Moderna vaccine and the technology underpinning both the Pfizer and the Moderna shots. So is this a way to create that broader access to mRNA products without colliding with the drug industry frontally? I'm also curious to find out about what you might say to critics who might say that this is a bit late in terms of securing access. Obviously, they might say that you had leverage much earlier in the pandemic and decided not to use it, and decided, well, not you specifically, but perhaps the US government. What do you say to that? Gray mentioned earlier that you see this as a model. Are you planning any similar collaborations in the future? What companies and what nations? And obviously, I understand why this is a big deal, but I think it helps readers very much to be as specific and practical as possible. Can you explain to us in the simplest possible terms what today's announcement enables the world to see in terms of mRNA products? Thank you so much.
Thank you, Donato. I think that was 4 questions, but and if I could just make a plea, this is really a scientific meeting. We've had there are many other aspects to this program: political, IP, equity, and so on. If we could try and keep the questions to the scientific ones, I mean, obviously I'm going to let Gray answer however he likes, but maybe leave the IP ones, for example, for another time. You have here available some really great scientists, and I think it would be useful to try and keep our remarks to the science. Gray.
Thank you very much, Charles, and thank you for your question. Yes, I think that this is a scientific colloquium, and that'll be the focus, but I can respond to your question. We collaborate with Moderna and other industry all the time, and those collaborations continue. We're not in a confrontation with any of those industry partners. We're in partnership with many of them, and we welcome additional partnerships with industry. This collaboration with Afrigen is a scientific collaboration to advance the discovery of new and the understanding of existing vaccines in use in low and middle income countries. As Barney said, our goal is the same goal that Afrigen has, which is to expand capacity to discover, to develop, and to run through the trials that Linda's designing new vaccines that will confront diseases where vaccines either don't exist or need to be improved. We do not see this as a confrontation between government and the private sector. We see this as another illustration of the essential nature that collaboration between the private and public sector, the essential nature of collaboration in discovering vaccines and even therapeutics that will save lives and that will enhance the lives of people around the world. So you know, we've very recently provided licensures to, as Charles mentioned, to the medicines patent pool, and we are eager to form partnerships with anyone who will utilize the intellectual property that we have developed through US government-supported resources. I think the federal government of the United States has been remarkably generous in supporting basic science that has led to discoveries that have then in turn led to products produced with private sector partners that have saved so many lives. Thank you.
Great. Thank you very much indeed. Great. I'd now like to turn to Sarah Jerving from DevX. You have a question?
Yeah, thank you so much. Dr. Terblanche, you mentioned this partnership reduces timelines. What are the new timelines specifically that will result of this partnership? And what is the latest on South Africa reaching WHO level 3 maturity? Thank you.
Sarah, thank you very much. This high impact acceleration is important for us to get GMP material ready for clinical trials. So as you know, this facility is a greenfield project. We have to get this facility fully licensed through SAPRA, our regulatory authority, to comply with all GMP standards to ensure that the vaccine that we put into humans are safe as in the first place and effective, of course, in the second place. The production of the pDNA material for our process here, our GMP process, will probably have a 3-month acceleration on the full process development and production of GMP material for clinical trials. And 3 months is quite significant in a greenfields project. Your second question, SAPRA. So I can comment. SAPRA is in the process to reach ML3 status through WHO. My understanding from latest feedback from WHO is that they're well on track to achieve that in 2022.
Great. Thank you very much. And I have an online question from Hushira Mamduk, I think, which is, and I'll probably turn to Barney with this. Won't the presence of lipids in the vaccine formula induce some health concerns such as autoimmune disease? Barney, would you like to take that?
Yes. Hello, everybody. This is Mushira Mamdou, the viral lab manager in Egyptian Drug Authority. I have
Hello? Did I read your question out wrong?
hello, can you hear me?
Yes, yes.
Yes, you read the question correctly. We're wondering about the health concern about the presence of lipid in the vaccine formula.
Yes.
This would raise a safety issue concerning autoimmune diseases, like what happened before with pandemic influenza vaccine. That also included a lipid. Yes, thank you.
Okay, thank you very much indeed. I will, we have such a fantastic collection of scientists here. I'm sure that many of them could answer this question, but Barney, why don't I turn to you first?
Yes, thank you for that question. The lipids in this, in the nanoparticle formulation are all degraded very rapidly. And in general, they are not antigenic. The pegylated lipid may occasionally trigger some things in people who have allergies to that type of lipid, but these are very rare. So on the order of 1 or 2 out of a million. That's why we ask people who get vaccinated to stay in the clinic for more than 15 minutes so that we can monitor for those. And those reactions are also easily treated, but it's also one of the things that could be available for innovation. I mean, I think the lipid formulation is something I think that will evolve, and I'm not aware of any concerns. I'm not aware of the plausibility of creating an autoimmune disease. I don't really see, because the lipids in general are just not antigenic. They can sometimes trigger innate immune responses. That's part of what creates the inflammation and some of the side effects after the second dose and things like that. With RNA is that innate immune responses are triggered, but that is not an adaptive response that could lead to some kind of autoimmunity. So I don't know of an example of that to give you.
Great, thank you, Barney. I don't know if anybody wants to add to that. I would guess that pretty much covers it. Great, thank you. Moga, you have a question. Would you like to speak it out, or would you like me to read it?
Thank you. Thank you very much. Basically, my question is about how impressive it is that South Africa reached that stage of development, and how the hub is doing training for the spooks. But how to get the spooks to reach at least similar development stage to the South Africa hub, and would the NIH, does the NIH has plans to help that? Thank you.
That may be a question for WHO. Is Somia still on the line? Somia, are you there? Well, the NIH will do its part. So provision of our technical expertise to our colleagues around the world is something we hold as a very high priority. We do have limited bandwidth. There's only so much we can do. We're not a training organization. We are not an organization that is a development organization, but to the degree that our technical expertise and our experts and scientists, as Barney said, can work with colleagues around the world, particularly through Afrigen, which will provide the training hub. We are training trainers who will then train others. To the degree that we can approach it that way, I think it will be a smashing success. And we're very happy to continue to play that part. Key to this will be the funding that those organizations receive from their own governments and from outside those countries. That is critical, and that is what has made South Africa the leader in this area, is that South Africa has long had support from DSI and from other, and MRC and others, to build their own capacity. So that's something that often people forget, how fundamentally important it is for their own research organizations to support themselves and not to rely solely on others, although certainly international donors and collaborators have a very important role to play. But for our part, as far as the science goes, we're happy to share everything and to do everything that we can to share expertise.
Great, thank you. Petra, would you like to add something to that?
Maybe Moga, just to add exactly that. Through the contribution that the NIH will make in terms of our process development, production of the pDNA, and the technology transfer, they will directly impact in the technology packages that will be transferred to 15 or 16 or more spokes to the mRNA Hub program. So there is a direct benefit that will go back to the spokes.
Great. Thank you. And I have a question here about if we want to vaccinate people really quickly, should we just be relying on using needles or other mechanisms that we should be thinking about, like dermal patches or whatever it might happen to be? I don't know, Gray, if you'd like to start with that, and then perhaps Glenda would like to comment.
I'd like to turn to Glenda, actually, on the importance of multiple.
Yeah, I mean, that's very important. And there are yeah, it's an important thing in terms of taking things to scale and also to getting into pediatric populations. And so there's some innovation in intranasal vaccines, particularly for SARS-CoV-2, and that will also have an application for TB as well as a possible modality. Other innovation is needless, using bioinjectors, needless designs. We don't need a needle and syringe for vaccine application. I think the most important thing is to find an effective vaccine and then to evaluate different modalities of administration once we know a vaccine works. And that's when innovation happens, because we do need vaccines that can be deployable rapidly and to populations like pediatrics in an easy fashion. So we are seeing that in the COVID development space.
I think Dr. Karine Bach might also have something to contribute to this. Karine, are you still on? Maybe NIH is also working in this space, and so we're very happy to contribute to those discussions as well.
Great, thank you very much. I'm going to take one more question and then going to wrap up this. So I'm probably going to mispronounce your name, so apologies. Hechmi Luzia, if you'd like to ask your question.
Yes, thank you very much, and congratulations for this program, and very ambitious program and very important for Africa. Congratulations also for South Africa, Afrigen, and the different institutions, research institutions and universities there to play the leader role for Africa. I forget to present myself. I am Hechmi Luzia from Tunisia. Of this big program on promotion of mRNA technology for vaccine development, etc. So I'm very excited, and it's very interesting, and we completely agree on what happens and the evolution of the program, and we are willing to contribute more in addition to receive the technology. I think that other spokes could also probably contribute to clinical trials, to evaluation, to some immunogenicity and other studies. So my recommendation is to think on how we can, let's say, contribute more in order to succeed in the development of the anti-COVID vaccine based on mRNA vaccine, Afrigen, but also probably other vaccine. And thank you very much.
Thank you for your question. Petra, I don't know if you'd like to comment.
Echmi, it's really great to have you on this meeting, and I think Sumia may also come in here. But absolutely yes, we've already started with the MRC and yourselves looking at West Nile, at Rift Valley fever, TB, and we need to build on that. I don't know whether Sumia wants to add, but we know that there's capabilities and competence and research innovation in the spokes to this mRNA Hub program that we need to optimally leverage to ensure that we reach successful milestones. We're in the young stage of the program, but yes, we will, and we will build on that. I don't know whether Sumia wants to add. No. Charles, do you want to add?
Thank you. Yes, I think unfortunately Sumi has had to go to another meeting, but I would just definitely say that part of this program is to encourage this collaboration amongst the spokes. And I think, as you just said, Hekumi, there's a huge potential here that we need to develop moving forward. So thank you very much indeed to all of our speakers, everyone attending, particularly the ambassadors. And in closing, I would just like to hand over to the Consul General of the US to South Africa, Todd Haskell, for a few remarks. Todd, over to you.
Thank you very much. As it's been noted, I guess this is a scientific colloquium, but they asked me to speak anyway. So I'm very pleased to have this opportunity to be here to commemorate the signing of this agreement between the United States NIH and Afrigen. I would like to start by recognizing all the hard work and effort that's gone into establish and launching this transformative partnership. Allow me to give a special shout out to my colleague from the US Embassy, our health attaché, Sophia Siddiqui. It's been a tremendous force, I think, for this partnership, but also other partnerships over the last few years together. So thank you for that. But also let me thank our colleagues at NIAID and our counterparts at DSI, at Afrigen, at Medicines Patent Pool, WHO, at WITS, and of course the South African Medical Research Council for their valued and long-standing partnership. This agreement marks an important technical commitment from the United States to support and to build on the future vaccine development in South Africa and frankly across the continent. I note that today South Africa has less than 20% of, or rather Africa has less than 20% COVID-19 vaccine coverage. I guess South Africa is closer to 31%. We in the United States were committed to vaccine equity, and we want to ensure access, and we want manufacturing to succeed, but it will need to be done by simultaneously addressing the disinformation, by taking science to the people and to building trust to make sure that we do get the vaccine uptake that we need to defeat this pandemic and future ones. I note that statistics tell us that South Africa has lost over 100,000 people, 100,000 lives to the pandemic. The United States has lost over 1 million, and the most of any single country. The pain of our nation is real. The pain of South Africa is real. The pain of the world is real. Throughout, we've stood with the global community. We've donated over 600 million vaccines around the world. We've provided more than $19 billion in health and humanitarian and economic assistance, and more than $1.7 billion to support vaccine readiness. We intend to increase our initial contribution towards the establishment of new pandemic preparedness and the Global Health Security Fund at the World Bank for a total of $450 million. The National Institute of Health has recently licensed research tools and the intellectual property for several COVID-19 technologies to the Medicine Patent Pool, through the WHO COVID-19 Technology Access Pool as well. Together, we stand by our commitment to working bilaterally, and I think very important to the Biden administration, multilaterally, to advocate and support vaccine manufacturing in Africa and in South Africa. President Ramaphosa has been a strong advocate for the continent in this effort, and we look forward to working with him, to working with other African leaders to make this a reality. This agreement is important for what it will achieve in scientific collaboration, but frankly, it's also a symbol of the unity as we move forward, as we face the next pandemic, and as we look towards the future. So thank you very much for your patience with this non-scientist, and I appreciate the invitation today. Thank you.
Great. Thank you very much indeed, Todd. And once again, thank you to you all for taking part in this, and to NIH and Afrigen for organizing it. This is an incredibly important program, and out of the disaster of COVID-19, this might be really such a great success that has emerged from this. So we should all really congratulate everyone who's part of this, and look forward to the future. Thank you.